Okay, hello, good evening, or good afternoon, everyone. Um, Joining us today on the RTC webinar series. First of all, we'd like to apologize for the slight long hiatus in our webinar series. We are not able to we are not able to um, have the continuous flow of the webinar series due to some reasons, but at the moment, I think it's rectified, and we will be bringing our own webinar series and our podcasts to you monthly or even twice a month. Joining us today is one of my teachers, which I'm proud to be here with him today. He's associate professor at the University of Ahmadu Bello University. He's really profound researcher, and if you look into his CV, I'm just kind of like really, really astonished. So he had his first and second degree at the Ahmadu Bello University Zaria and proceeded to South Africa, where he had his terminal degree, all in the field of biochemistry. But specifically, his research is much focused on tag of molecular biology, biotherapeutics, drug design, and that is especially targeting trypanosomiasis, malaria, and type 2 diabetes. So our speaker, who is in the person of Dr. Muhammad Awal Ibrahim, had several fellowship, which include in South Africa, in University of Pretoria, and also in Japan, which is just of recent from 2019 to 2021. He had more than 120 scientific published articles, which are available, with a Hench index of 33 and of an item index of over 70, according to Google Scholar. So he had supervised more than 30 MSc students, with 3 graduate student, PhD students, and has taught just all over the world in the name of science, from all over the continents, Europe, Asia, South America, North America, Africa, which is just home. So, for anyone that is really interested in the field of drug discovery of molecular biology, this is really an interesting talk. You can listen to us live, which is available on our social media handle at BioRTC, and our Facebook, on Twitter, and on X. And you can post your question on those platforms. And at the end of the session, at the end of the presentation, we'll be able to read out the questions you have sent through the chat box for our speaker to answer it to them. And even after that, this webinar will be available on demand on our social media handle and also in our website at BioRTC, www.biortc.com. So just a brief introduction for those who don't know us. BioRTC is the first and only open access core facility in Nigeria, housing state-of-art equipment, and the main objective is to improve the science in Nigeria and in Africa. And this webinar series and podcast is to entice and to bring those performing research, those in research, close together, improve collaboration, entice the younger generation into science, and make African science in such a way it will benefit the global knowledge. Thank you very much for joining us today in the studio. And without further waste of time, I would like to give the floor to the speaker so that we will hear from his presentation. Thank you.
Okay. Thank you, Greg, for the introduction. I really appreciate. Uh, Also, let me start by thanking the BioRTC management. Sorry,
Yeah,
it's my flight moving.
yeah, it is. We
Okay.
can see, even though it's not on slideshow. I don't know.
Actually, I've converted it to slideshow. I hope I'm not
Okay,
having another embarrassment.
but I think it's clear. We can see it.
Yeah, but not on the slideshow. That's good.
Mm.
Anyway, let me thank the management of BioRTC for inviting me for this webinar series of the center. I really appreciate. And my talk, like I've said, I wanted to talk about paradigm shift in drug discovery, especially from the perspective of, especially from the perspective of antitrypanosomal drug discovery. Uh, but Zaid, I think I'm having some challenges. I can see my slide moving from this side and it's not moving from your side. I, I'm moving it, but it's a bit stagnated there. I don't know.
Yeah, that is such a pity. I don't know if you can just send it to me, then I will present it. Then I don't know.
Okay, while we do, I will do that, and as I do that, I will also try some other notes.
Hmm.
Sorry about that.
Sorry, viewers, for the slight glitch we have in the technology.
Please put this thing for granted. I'm trying to send it to you.
Okay.
Okay, is there?
Mm, okay. Received.
Okay, sometimes. Sorry.
Okay. Okay. Why am I also having this discussion? Is this?
I'm sharing my own screen. Can you see?
Okay. Yeah. Yeah. Okay. I can see it now.
Let me try and see whether that will work better.
Okay.
Yeah, I think this works.
Okay.
Okay. Uh, we are really sorry. I think we have taken the technology for granted.
Yeah.
Yeah. Once again, I really appreciate the presentation by RTC Management, chaired by Dr. Mahmoud Bokar Maina. Thank you. My presentation is going to be on the paradigm shift in drug discovery within the context of anti-trypanosomal work. And the basis for this is because I actually want to call our attention, especially those of us that are young and upcoming, to the changing, to the very fast change in research landscape across the world. That is actually what informed my decision, choice of this topic. That I want to talk about paradigm shift of African biomedical scientists within the anti-trypanosomal drug discovery strategies. I want to use a case scenario, particularly of myself, to show our upcoming scientists that there is need for a paradigm shift, and that is the only way that those comforts in Africa can make very impactful and meaningful research that will eventually be useful to both Africa and the entire global scientific community. So, for most of us that are in the biomedical science arena generally, we, as far as drug discovery and development pipeline is concerned, generally we work in this area at the at the earlier stage, that is starting from target discovery to lead discovery, lead optimization, and at best, preclinical trials. So, for some of us that are in biomedical research, we are largely focused in this area, and that is why most of my presentation will also be focused on this aspect. And before I go further, let me also give us general overview of the current strategies for drug discovery that is topical and quite applied in even big pharmaceutical industries. The current strategy for drug discovery, this is the workflow, generally from target disease and target disease and also target gene, and then from there the target protein is identified, and all this will revolve around the target protein is identified, and then the target protein is also, after the identification, structural biology of target protein proceeds, and issues also. This generally involves recombinant DNA technology and a lot of structural biology work. And from there, hit identification, and then lead compound, and then from there, the candidate is being identified. Now, this aspect is one that actually involves uh, basic some of the testing that we do, especially in the African continent, as far as the diseases are concerned. But we can see that despite this workflow, there are so many other things that are neatly integrated into the workflow, involving from pharmacokinetics, pharmacology aspect, bioinformatics aspect, genomics aspect, and so many other. In fact, generally the omics are all integrated into the workflow. And so this is now the modern, or let me use the word, modern drug discovery strategies that are really applied, especially in Nigeria. And I usually say that, despite the fact that this is the now workflow for drug discovery as applied in the global scientific community, but in Nigeria, we actually tend to look at drug discovery as being almost equal to medicinal plant research. Most of us try to look at drug discovery as if it's equal to medicinal plant research, and that it's actually not so, as I've shown us earlier. The world has moved far ahead, and that is the trend that is being applied. Now, so because of this, I want to, and it's because of this, I don't want to say misconception, but because of this general notion, that is why I want to now, I thought to share this seminar to talk about paradigm shift, meaning that what I'm saying is the fact that this our notion of drug discovery to be almost equal to medicinal plant research within the Nigerian biomedical research community should be changed. There is need for paradigm shift from this thought and this action. And that's why I want to now cite some examples. Recently, I tried to visit the official website of journals, professional journals within the biomedical field in Nigeria. And let's look at this. We can see that the first one, you can see what I realized is that nearly more than 50% of the research that are there are actually medicinal plant research. And so I now, if you look at it, this is one of them. I now just sample them, and I'm going to share some of the samples with us now. This is an example where the issue of uh, Xylopia ethiopiaca here. This one, Carica papaya. The next one is Nigella sativa. Then I also went to another professional body. I'm going to give us the professional bodies. This one is aqueous leaf extract of Jatropha tangerensis. This one is ethanol extract of Ricinus communis. This one is coconut. So many. Like I said, more than 50% of most of what is there is actually revolves around medicinal plant research, and that's why we see it as if it's synonymous to drug discovery strategy, and that is the paradigm shift I want us to start looking at. Now, from this, look at this. The first one I've shared is actually from the anatomical sciences. This is from the official. These are the articles that I retrieved from the Anatomical Society, Journal of Anatomical Sciences. That is the official journal of Anatomical Society of Nigeria. Now you can see as if the works are come from the same discipline. Now the next one is from physiological sciences. This one is from Nigerian physiological science. You can see both anatomy heavily investing in medicinal plant, physiology heavily investing in medicinal plant research. Now the next one, look at this one, antioxidant activity toxicology of aqueous extract of Azanza gyncaena. The next one is also acute toxicity, acute and subacute toxicity of the same plant, aqueous extract, then Ramphylum 12, and then the next one is Pterocarpus milbredi. Now that's, you can see medicinal plant. I'll also share with us which field is this. Now look at this one. We see sedative anticonvulsant, also ethanol extract of Legionnaria brevifolia. The next one is fruit extract of oil palm. The next one is Adansonia digitata. Now, the first set, as you can see, is actually biochemistry and molecular biology. And the other one is pharmaceutical sciences. So you can see anatomy, physiology, biochemistry, pharmaceutical sciences are all heavily invested their energy and their research prowess onto medicinal plant research. And this is from this week. This is also another one. You can see Anagenesis leocarpus. You can see medicinal plants, and we can also see even Moringa oleifera. This is microbiology, and I'm sure if you do this for parasitology, for even some aspect of medical lab science, for zoology, for botany, for pharmacognosy, and so on and so forth, the picture is essentially the same, and that is actually what leads to Nigeria having a very high output of medicinal plant research. For instance, in 2018, up to 4,000 articles in medicinal plant research from the country. This is quite huge. This is quite huge. And someone, we may say that it's even commendable. It's huge and quite commendable to a certain extent. But the point is, what product are we generating from this research? What are the products emanating from this research? 4,000 articles in a year. What are the products? Presently, I know we can all guess the answer as Nigerians. We can all guess what the answer is. But what I want to tell us is the fact that the problem why we are not having products, even though I say we can guess the answer, but we can guess the answer is that there are no much products. From 4,000 articles in 2018, I don't think uh, there are no much products generally from medicinal plant research. It's actually because of the fact that we keep repeating and repeating and repeating the same thing. The same plant will be investigated over hundreds of bioactivities and across specialties, and then at the end, it will now be started all over. And that is what I'm showing from one to, if it is one plant, a plant will be investigated by a particular discipline, the next one, the next one, up to 4. Then there will be reverse. And that's what has been going on in the area. And this also, apart from being discipline specific, this repetition, it also has to do with time. You see, for instance, some research activities will now usually start from a particular technique, a particular time, and then after some years, you see as if uh, uh, the scientific community goes back. That has been a bit ongoing if you go through the literature, and that is what I think, in my opinion, has dragged drug discovery in Africa, especially in Nigeria, a little bit backward. It's not moving as fast as it's shown because of some of these repetitions, and that is why there is serious need for the paradigm shift. So, like I said, there is need for paradigm shift, which will allow us to move forward and progressively forward. But the paradigm shift always comes with its own challenges. It needs a lot of technology. We need to evolve. We need to a lot of diversification, and we also need some disruptive innovations for us to have a meaningful paradigm shift in the research. I'm going to cite a number of examples. One approach for that we can exploit in pursuing this paradigm shift that uh, I'm, 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 I'm saying is that first we need to break the barriers. You can see from what I've shown you, anatomical anatomists are doing their own, biochemists are doing their own, microbiologists are doing their own, and so on and so forth. But if we are to move forward, the paradigm, one approach for this paradigm shift that will allow us classically move forward is that we have to break those barriers. We have to break the barrier of working in silos, pharmacology doing its own, pharmacognosy doing its own, biology doing its own, parasitology doing. We need to break those barriers. I think, in my opinion, that is the first approach that has to be used. We need to break those barriers so that we start achieving multidisciplinary, interdisciplinary, and also transdisciplinary research activities. I think that is one way. And at this point, I must commend the BioRTC for a very job well done, because BioRTC, even though has been largely funded, the founder has a days in anatomy, but you can see how everybody in the biomedical science field is being taken along. That is one way to break this barrier so that we can make meaningful contribution to the world of science, and we can also use biomedical science to solve our local problem within the Nigerian community. Another approach is personalized effort, and I think I will try to say much here. Like I said, the personalized effort also involves a lot of challenges, a lot of technology. You need to disruptive innovations and so many other stuff like that. And I will try to use some of our previous examples over the years, in the last 15 years or so, to show how this personalized effort can be, how paradigm shift can be achieved through some of this personalized effort. That's what I want to show. And also, at some point, I will use the same scenario to show us a multi-layer paradigm shift. We can have different shifts, but eventually the shift there have to be shifts that will allow us to propel forward, that will allow us move forward as a community, move forward as scientists. So also, before I go further, I want to really call our attention to my colleagues, especially the young scientists, that, like I've shown us, so many duplications, so many replications, so many repetitions. As we grow into the scientific arena, we should, as much as possible, try to run away from that. At every single point we move ahead, that is the only way that we can move forward, and that is the paradigm shift that I'm actually trying to call our attention to. So the first one, I will be focusing today on what we have been doing on African trypanosomiasis. I want to use this case study to show how we were able to make some modest paradigm shift in this area of research. So that perhaps we hope that this can be a basis for our colleague to use this model so that we can advance our individual research effort and consequently make meaningful scientific contribution to the world of science and also to solving uh, problems in our respective communities. Now, African trypanosomiasis is a very serious disease affecting the African continent. We have two forms: the human African trypanosomiasis, and we also have the animal African trypanosomiasis. African animal trypanosomiasis at the moment is much more prevalent. I think at a point accounting for up to 41% of the animals in Nigeria are being affected by African animal trypanosomiasis as far back as uh, uh, last last 10 years or so. So that is why we have been working very hard to see how we can develop new drugs against the disease because the current drugs have so many therapeutic limitations. Now, the the first example of a paradigm shift that we have tried to champion is the paradigm shift within the medicinal plant research. So what I'm just trying to say is the fact that even within the medicinal plant research, we can make meaningful change, we can make meaningful shift so that we avoid moving from one point A to point B and from point B to point A back and forth. That is what I want to also demonstrate. Perhaps this can be a model for some of us to utilize this to also move forward, especially like I keep saying for the for fellow young scientists. Now, in 2018, this somehow about one of my first articles. In 2018, sorry, 20 in 2008 to 2010, we have actually investigated the antitrypanosomal activities of a number of plants. This is one of them, Kaya senegalensis, and that was 2008. And then we also investigated another plant. This is Pannonia blumidis, and we also investigated another plant, Senna occidentalis. So you see, we are also repeating, just investigating antitrypanosomal activity of plant A, plant B, plant C, plant D, and so on and so forth. These are just examples of some of them that I just picked to show us here. But the point is, after doing this, let me say this round, we started thinking of a paradigm shift. Do we continue of just investigating plants left, right, and center, just changing plants today, tomorrow, next tomorrow? I just we at that point we just thought that is not the way to go. There is need for paradigm shift, and that is the first thing I want to call our attention to. Then we now went back to one of the plants, the Chias senegalensis. Like I've said, I've only shared 3 plants, but we have investigated so many and also reported them as such. But we just realized we are just repeating, just changing plant. Then we now pick one of the plant. We said, okay, the next thing is how do we now identify the chemical constituents that are responsible for the activity that we have observed there? And we now reported observed 2 compounds to be the compounds are responsible for the activity. In fact, we even tried some synergistic study to see how we can now improve the bioactivity that we have realized with the Kaya senegalensis. But all this to say, I'm just trying to summarize because I'm just trying to show this as a model. I'm not going into technicalities and also the science and define this specifically. I'm just using this to demonstrate that shift is needed and shift is possible. Now, we tried this and it eventually failed. After the basic pharmacology, I think this one, not much activity with, okay, good activity. We also tried to understand the chemical constituents. We identified the chemical constituents, but there was a problem, especially with toxicity. We now try to see how do we now reduce the toxicity. We tried some synergistic study, it actually failed. After this, we even got the pure one of the pure compound and also tested it. and we realized that the activity was not as potent as we observed. And this paper was published in 2021, and you can see in short, from 2008 to 2021, we've been working on some plan moving forward, but eventually this is where we were. And I would say that this is not where we want to be. And very importantly, because of the fact that the activity that we eventually ended up with is not something that is quite remarkable. And that is one deficiency of the approach that we are taking in medicinal plant research. And that is why we need some paradigm shift. So we can see even within the medicinal plant research, we have made some shifts. We started from basic pharmacology, we moved to identification, and then we moved to including some computational chemistry. Now, at the point, we also said something like I've earlier said, that there is a problem. If we keep repeating the same thing, we are not going anywhere. Then we started to go into literature to see what is happening in the literature. Within the literature, it was shocking to us as of then, that was in 2014, to realize that for this antitrypanosomal study that we have been studying as of then, 264 plant species were investigated for antitrypanosomal, but there was no much progress. Antitrypanosomal, and with so many repetitions, as of then we realized 264 plant species from the African continent, alone, they were investigated, and there was no much progress. There was no drug that has been developed, just revolving around the plants, move, just checking the plants and throwing and leaving them. Then eventually only 48 compounds were isolated, and in fact only one of them as of then was investigated in vivo. So we now thought that is now a gap that we will bridge. So this now forms the basis of our next line of research. And I will just rush through the research. We investigated a number of compounds. These compounds are actually, like I've said, from this literature, we now thought it wise that now if we are to move forward as in the African if we are to move forward in the antitrypanosomal drug discovery within the African continent, then this is the way forward. The way forward is some compounds are already identified. The next thing is we can now take them further. That is one step. This has now saved a lot of time for us without a lot of time for us. So this now forms the basis with hope that we can now take most of these compounds into preclinical trials, and that's what we exactly did. So we tested one of the compounds called ellagic acid. We both basic pharmacology. When I say basic pharmacology, I refer to basic testing of the antitrypanosomal activity in animal models. And then we also try to use basic enzymology to see how it interacts with a very important enzyme in the parasite called the trypanosomal silage. We also use computational chemistry to see how the enzyme and the compound interacted. Now, well, of course, the work was published. Then we also did something with another compound, sigmasterol. We also did nearly the same thing with another compound, acetone. We also did something with beta ion. But in the case of beta ion, we also tried to shift a bit because we have realized yes, most of them have potential activity, and we also did the enzymology aspect. The next thing is molecular biology aspect. So by 2020, we tried to also advance some of our findings. So sorry, some of our studies to venture into molecular biology so that we can get more mechanistic insight into what is happening. And that is how that's one of the things that we investigated here, where we also included the effect of the compound with transalidase, especially T1TS3, T1TS4 gene expression systems, gene expression. So we also investigated that. But by and large, we also did some computational studies and also in vitro study with another compound, beta cytosterol, and we also did another one with another plant called eugenol, where we also look at how it affects the trypanosomal, the proliferation of the trypanosome, and very importantly, we also see how it also mitigate the expression of transthyretinase gene in the parasite. Now, with this, despite all this effort, but we can see clearly so that we are we it's just we are just largely changing so many compounds. We keep changing compounds, and essentially, no much progress has been done apart from progress in terms of sophistication of the experiment, apart from sophistication in experiments are also and also using modern techniques. Those are the paradigm shift that we have created within this antitrypanosomal drug discovery from the perspective of medicinal plants. But we have not done much. What should have been seen is by now, I think we should be able to see that this is some of the derivatization that were done. This is now the heat that we have. This is now something that can go into clinical trials, perhaps. But because of the limitations of African science, we have this. We have to rigmarole, play around with compounds. As you can see, we tested. These are also so many examples. These are also some of the examples. We have tested a lot of others. So before I go further, I want to call our attention to fellow colleagues that we can see even within the medicinal plant research, we are also able to make some shifts. But the shift is not enough. It's not sufficient to make to be very very impactful as we hope to. So I want to use this medium to call my colleagues, especially those of us that are young, that despite the challenges, we should try it even if we work in the area of medicinal plant research. It's a very good area to work with, but we should try to be advancing, try to be advancing from one point to the other, not to be changing plants or not to be changing compounds, but to be moving from one point A to point B to C, D, and by after some time, certainly we will arrive at point Z. And so this is one of the limitations. And because of this, of course, this is a summary of some of our findings. This is now the shifts that we have created within the medicinal plant research. In summary, I want to provide us with this shift. First, you can see that we started with crude extracts and we moved to pure compounds. And within the crude extract, if you can remember from all that I've presented, that we work on basic pharmacology, then move to chemical identification using analytical instruments, including GCMS, LCMS, and others. Then we move to pure compounds. Within the pure compounds, we also tested the basic pharmacology. Then we shifted to enzymology. We shifted to bioinformatics and computational chemistry, and we shifted to molecular biology. So we can see that over the years, in the last, starting from 2008 down to around 2018, one decade, we actually made shifts within the medicinal plant research. And this is what I want to call our attention to, that even within the medicinal plant research, we should endeavor as much as possible to be moving forward, not to be revolving, as I've earlier shown us so many examples from some of the journals that I've shown. So this is a shift that we have made in 10 years within medicinal plant research, which I hope will serve as model for fellow colleagues to also peruse. Now, as I've said, we have made so many attempts with so many shifts over the years to find potent lead compounds against an animal African trypanosomiasis, but clearly, as I've said, the success was limited, and so we need another paradigm shift. So you can see if there is a word like micro shift and macro shift, we have made small shifts within the medicinal plant research, as I've earlier demonstrated, but that was still not enough. We need, let me say, a complete paradigm shift now. And that's why I'm calling this one a paradigm shift, while the other one call it basic just shift. We need a complete shift. And now, when we were thinking of this paradigm shift, because we realized that that was not going much, that was not taking us very far, we now went back to the classical drug discovery approaches that is a strategy that is being applied by the big pharmaceutical industry and by the global scientific community. Now we look at it and we said, okay, now let's start from this point. And that is what now take us to one of the techniques that is being applied by the big pharmas, the so-called fragment-based drug discovery, which involves high throughput screening and the use of uh, screening collection of very small molecules which we call fragments, and we subsequently merge, expand, merge, and link them. So in this technique, usually low molecular weight chemical fragments are selected on the basis of ability to bind to target. I'm going to demonstrate that. Target of interest or inhibit it in a functional assay. And then eventually we use those fragments as basis to build more complex lead series, and then those ones are subjected to high throughput screening. Now, in short, there are three stages in the fragment-based drug discovery. We have the fragment generation stage, where we apply the rule of three and not the conventional rule of five in the selection of fragments. And the fragments usually come from so many sources, from commercial databases, from Chebi, Zinc, or from pharmaceutical industries, Novartis, Kishida, May, RO3 fragment library, IOTA, Life Chemicals, so many others uh, are available that produce fragments that are quite useful. And so I will also show how we use some of these fragments in our studies. Now, the next stage is fragment hit identification, where we investigate the ability of the fragments to bind to the target using direct binding assay, or using subsequently we also try about physical screening, including NMR based screening. And then if successful, we try to use crystallography based approach, especially X-ray crystallography, to see how we can get a complete picture of fragment binding to a particular target. I'm going to show us. Then eventually the fragments are linked, and this approach has been used, like I've said, by big pharmas, and so many drugs have been developed. And therefore, we now ask ourselves whether we can report this success in African animal trypanosomiasis. We selected a target, very important target for the survival of the parasite in affected animals, so-called GPI phospholipase C, which is very important in antigenic variation. It allows the parasite to contribute to antigenic variation by allowing the parasite to change its variable surface glycoprotein so that it can escape the host immune response. So we selected this, this, this target. After selecting it, we obtained the synthetic gene. We cloned the synthetic gene onto plasmid and eventually digested the plasmid, got the construct, the plasmid gene construct. You can see the ET15B with containing the gene of interest, that is the trypanosomal phospholipase T, and eventually after successful cloning, we also tried to express the protein, and the protein was successfully expressed. This is the profile, the SDS patch analysis of the recombinant trypanosomal phospholipase T after subjected to purification using affinity chromatography, that sister column, and ion exchange, and also gel filtration using fast protein liquid chromatography techniques. And after this, we now try to check. These are all stages and strategies that are now used in drug discovery, especially in fragment-based drug discovery. Now, after purifying the protein, we needed to confirm the oligomeric state of the protein, and that's why we now subjected it to circular dichroism. Then, very interestingly, it gave us a very good level of cooperativity, showing the protein is in a very good oligomeric state. Then, having confirmed that we had an intact protein, we now subjected the protein to high throughput screening, where we use the Maybridge RO3 diversity fragment library, and also some fragments from Kishida Chemicals, where and used 874 fragments and subjected them to inhibition studies, high throughput screening. Eventually, we realized that among all the 874 compounds, rather fragments, only 65, based on our set criteria, only 66 actually pass our selection criteria, where our selection criteria was at least 40% inhibition of the recombinant protein. So this 66 primary hits compounds, that means we are now sure those compounds are able to inhibit, in a functional assay, they are able to inhibit the recombinant trypanosomal phospholipase C. We now subjected those 66 hits to a technique called the water ligand observer gradient NMR spectroscopy, the water log Z NMR spectroscopy techniques. And the basis for this is that we were trying to determine the direct binding of all those 66 hits to the recombinant protein, because it's been known by in fragment-based drug discovery, biochemical assays are generally not very much reliable because they produce so much false positive. And one way to remove false positive is to now switch to biophysical assays. And this is actually what the big pharmas also use. They use generally biophysical assays to solve some of these research problems. So we subjected this 66 primary hits to water log Z NMR analysis, and interestingly, only 10 were able to bind directly to the recombinant protein, indicating that the remaining 56 are actually false positive. And you can see this how important this is, how important the biophysical techniques are in trying to remove false positive results. So after this, with this now, with the waterlogged NMR technique, we are now very sure and very confident that we have 10 fragments that are able to bind to our recombinant trypanosomal phospholipase. Now we are sure about it. The next thing is, this is now the structure of the fragments. Then we try to see how each of these actually bind to the protein. Because, like I've said earlier, in fragment-based drug discovery, we now identify fragments. The target is to now link or merge fragments so that you can have a lead compound. That is actually the target. So we now have fragments, 10 of them. The next thing is how do they now bind to the recombinant protein? And if they bind, how do we now match, like now create or link up relevant ones so that we can completely uh, have a drug, a lead compound that is able to completely inactivate this protein. So we now, of course, the next thing, one approach, like I've said, we will have used also X-ray crystallography. I will show us some of the data later. But we also use computational analysis where we saw how they mostly bind. But the most important is this one, where we saw that some fragments, especially fragments 6, 7, and 9, bind somewhere up. Then 4 can serve as linker, and then 1 and 8 bind. Luckily, they all bind around the active site of the protein, meaning we can now, if we want to create a very successful inhibitor of this protein and that can serve as a successful drug, we have to now link fragment 6 with 4 and 1, or 7, 4 and 1, or 9, 4 and 1, or 6, 4 and 1, or 7, 4 and 1, and so on and so forth. Essentially, either of these fragments 6, 7, and 9 will now be linked with fragment 8, and always fragment 4 will be linker. This is quite very interesting, and with this we are now on the path of generating this lead compound. So we can see that with this we are able to shortcut this, and this was done in just 2 years. So we are now able to, all that we have achieved in the last 10 to 12 years, we are now able to achieve it in less than 2 years, and with better, more precise information, and more and with more precise information and more effective. Of course, this was we also try to subject them to X-ray crystallography. Some of them are from the crystal, but were not successfully diffract. So eventually, the X-ray crystallography was not successful, and in fact, that's why we actually adopted the computational means. Also, we tried cryo-EM electron microscopy to see whether cryo-electron microscopy to see whether we can actually solve the structure of the recombinant trypanosomal phospholipase C in complex, either free or in complex with our fragments. But unfortunately, also that did not yield result. And eventually, some of this information have been published. And our final note, because of time I'm rushing, you can see that we use the, let's say, modern drug discovery strategies, especially the use of recombinant DNA technology, bioinformatics, biophysical methods. We have shortened the pathway for antitrypanosomal drug strategy from nearly 12 years to just 2 years. So we can now see this is a demonstration that if we can now make we need those paradigm shifts to shorten our uh, our research achievements, or rather, not shorten, to facilitate our research achievement and at the same time make quite impactful findings. So in conclusion, I want to say that in spite of our consistent shift and progressions in the area of medicinal plant research, we could only achieve limited success in the area of antitrypanosomal drug discovery. But when we made a paradigm shift towards, especially towards molecular biology tools, including recombinant DNA technology and fragment-based drug discovery, we are now able to discover novel hits with potential targets, with good targets against trypanosomiasis. And so our paradigm shift has led to a shortened period of anti-trypanosomal drug discovery, in addition to improved precision and effectiveness of our drug candidates. So this is now what I want to share with us this afternoon or morning, as the case may be. And once again, I want to call our attention the attention of all colleagues in the biomedical science field, ranging from anatomy, physiology, biochemistry, microbiology, pathology, all of us, that we need this paradigm shift. Even if we are to stick with the medicinal chemistry, medicinal plant research arena, we need some paradigm shift. We need some shifts within those arenas, and otherwise we can also, if opportunity presents itself, we can make a complete paradigm shift. And I think this is one model that we can use for some of us in Africa to make impactful contribution to the global scientific community. So I acknowledge all this, my founders over the years, and I thank the BioRTC for the opportunity. Thank you.
Thank you very much for this exciting talk. I'm mostly immersed in the in-depth and how interesting the kind of the angle of um, of the presentation that I just got lost and I even forgot about the time. But it's really interesting.
Thank
And
you.
on this particular note, I just want to make an announcement, and I'm really because I have to even tweet about that one particular slide of yours that highlighted the contribution of BioRTC as a major stakeholder in kind of contributing to the paradigm shift. Yeah. So that is particularly interesting. And being a core facility, I know the major problem affecting African scientists, both in diaspora and those in Nigeria, is the issue of funding.
Yeah.
We usually don't find ourselves in a stage where we'll be able to apply for funding. We don't know how to access the funding both within and international. And the presence of BioRTC as a core facility and is open access, it means our arms are really open for any scientist in any biomedical field, because we have a lot of cohorts within the biology. And the person wanted to apply for a grant, be it international or national, the person can reach out to BioRTC, and BioRTC will serve as a host, because the most important thing is having the base institution. Okay, I'm applying to do this kind of work, like all the complex work the prof presented, is definitely not done in Nigeria because we don't have the means to do that. And if you don't have the means, it means you cannot apply for grants. You can never be an independent researcher. You have to just probably go, maybe live there, not in Nigeria. So one of the vision of the RTC is for any researcher in Nigeria can reach out to by RTC. You can check out our website on www.rtc.com. Look into send us that okay, you want to apply for social grant, and the center will support you with all the necessary means to see that the grant is fruitful, so that you're able to come to do the work in Nigeria. Because trypanosomiasis is an African thing. Mosquito malaria is African thing. Like no one understand it like Africans. So we are the people to solve our problem. Thank you very very much for this interesting talk. I really really enjoyed it, and this webinar is available on demand anytime. You can access it on our social media platforms at biortc dot at biortc BROIRTCNIG on YouTube, Facebook, or X. You can just wait it on any time. So without taking much of the time, I have my questions, but I see we have some questions from the audience as well. And in the interest of time, let me just go. We have so hmm, so that is this one question from Mohammed Mustafa. What factors put medicinal plants at greatest risk?
What factors put medicinal plants at greatest risk? Uh, Even though it's not very clear to me, the question is not
Yeah.
very, but let me just say that I'm not saying at the same time that medicinal plant research is something bad. It's something very good, very
In,
good.
mm,
But the point is, what I want to call our attention is that we are repeating the something, I'm not moving forward. That is just what I'm just saying. And that is, I think, the summary of what I'm saying. It's something very good. I think let me correct that notion. At any way, medicinal plant research, I have been an advocate of medicinal plant research, a very serious advocate of medicinal plant research in Nigeria, because that is one of our strengths. But how we pursue it, in my opinion, is what is wrong. Like I've demonstrated severally in the course of my webinar. We did it, we keep repeating without making serious effort to move forward. And that's why I gave example of a, a shift within, I demonstrated a shift that we try to make within the medicinal plant research, and I also demonstrated a shift that we made within the antitrypanosomal drug discovery, but out of medicinal plant research. So it's a multi-layer shift that I actually tried to demonstrate as model that some of us can use. So in short, I want to tell my friend Muhammad Mustafa that there is no much risk, even though, like I said, the question is not clear. There is no much risk, there is no problem with it. Let me use that word, but it's the way we pursue it is that we need that paradigm shift. I hope this answered the question.
yeah, I'm sure it does because yeah, exactly. I think this is one of the questions that really popped to me while I was on in my undergrad. I was asking, okay, we have research going on for like couple of decades within ABU Zaria, and I said, what is exactly that we have produced? That's a layman question of thinking, like because I wasn't aware of anything apart from that research is about discovery at that moment. But I was like, what have you produced? And the presentation is just the nail in the perfect place because we just keep repeating things. We have and yeah, I think it's a very, 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 very. Is this particular thing is a discussion we will have. We can discuss it for the next entire hour. Yeah, but there is one interesting question from Hafiz Mohammed. He said, "Thank you for your presentation. One thing I understand is that changing the paradigm should require more tests and more techniques, which means more funding. How can we change the paradigm shift as Nigeria, MSc or PhD student? Most Nigerian students have no funds."
Okay. I will. That's why I actually give like I said multi-layer. How we shifted within the medicinal plan, how we shifted out of medicinal plan. Uh, I will tell you. I also expected question like this, but I want to tell you, yes, there is no funding. That is the truth. That is the bitter truth, and we have worked a lot without. We have suffered a lot without funding. But research, I attended one of one of the topmost scientists, or rather cited scientists in the world. I've interacted with him once in my life. He told me something which I always remember. He told me that good scientists do not need, whether you agree with it or not, but I want to say it. Good scientists do not need sophisticated equipment, but you only need your two good eyes with some wires that are connected to your brain. That is what he told me. I will never forget that. That a good scientist do not need sophisticated equipment, but what you need is just two eyes with some wires that are connected to your brain. And if your brain is good and you can see, you should be able to think and solve a problem without sophisticated equipment. Like I've said, whether you agree with his thoughts, he's actually not an African, he's a European. Uh, whether we agree or not, that is a subject for another discussion. But what I'm saying here is that yes, there are challenges. There are challenges, no funding. But even with those challenges, rather than to pick, let's say for instance, one of the paper I've shown us is somebody work on antibacterial activity of Adansonia or Moringa oleifera. That's one example. I've shown. It's one of the examples I've shown you. And this is, like I've said, this is the latest issue in the journals. Latest issues I was able to trace this morning. Moringa oleifera, antimicrobial activity of Moringa oleifera. You see, for instance, we know Moringa oleifera has antimicrobial activity, and in fact, even the microbe that was tested, if you check the paper, I can show us, is E. coli and Staphylococcus aureus. You can see phytochemical and antibacterial properties of Moringa oleifera leaf extract on E. coli and Staph aureus. For God's sake, rather than to do this, we know Moringa oleifera has been investigated for antimicrobial generally for years, for decades. In fact, some countries have gone ahead of to even standardize and is being formulated, even though for other purposes. But it's well known. So rather than to do this, why don't you create a paradigm shift to say to do something that is one step ahead of this, rather than to repeat the same thing? So you see, and I'm not sure, but I believe this set of people did this study without funding. They did it without funding. So rather than to do this without funding, it's better they think of something without funding that will be one step ahead of this. If our my fellow students will keep moving one step, if you take, rather than to also do Moringa oleifera, don't take another plant within the Moringa oleifera, maybe something either mechanistic study, or try to now say you are now standardizing it and testing. Do something that will now make it forward, even within the medicinal plant research. So I'm saying that what I'm saying in essence is the fact that yes, there is no funding, but we can still make some shift rather than to be rigmaroling around the same point. And to tell you the truth, I think most of the earlier papers I've shown you were largely executed without the funding, and you can see we were able to make some modest shifts, progress without the funding. So that is what I'm trying. It's quite challenging, I must say, but it's doable. That's what I'm saying, and I think it's doable, and it's far much more better than rigmaroling and repeating the same thing. So those are my thoughts. Thank you. I think we can hear you. Say it. You are muted.
Oh, sorry. I just I think that really answered the question perfectly. And this, as you were presenting, is exactly what I've been thinking. What is usually the problem? Because when you think of hard work, when you think of the way Nigerians think and even African scientists think, when you find them outside the realms of Nigeria, find them to be doing really well. But within Nigeria, you just don't understand what is really going on. Are people just looking for easy way out or just to survive? Like they don't really care about the science. It's kind of like, and probably we will have to host you again for another podcast so that we discuss this issue and we don't have to present anything but just tap from your immense years of experience and what you have like really realized among in the mentality of how people such as think.
That's very okay. But like you said, that's a very broad. That would be a very broad topic. Those
Exactly. We'll have an hour for that discussion probably.
and the truth is, I was able to, by God's grace, to not seriously, but as I've said, the earlier papers there was shift, but the shift was not very meaningful, I must say. But there was shift. That
Mm,
was there was shift, and if one can make shift, then another. That is just one individual. If hundreds of people will make similar shift, no matter how infinitesimally small it is, certainly after 10 years we will see it manifesting and
exactly.
contributing in the arena of
Yeah.
global scientific arena.
Research is about community, it's about collaboration, it's about
Yes,
a lot of scientists pulling together to solve problem.
together. Yes. So
Yes.
that's actually my thought. Yeah. Next
Yeah.
question.
And that is, yeah, this really is a sequel to that question. And I think it's really interesting we have this up. So it's from Uthman Abdullah here. What is your advice to young students like undergraduate whom like to specialize in bioinformatics and drug discovery in future?
I think the advice is straightforward. What one of my mentors, Professor Brian Onions, advised us is to read. He always tell us just read. I will tell you, Mr. Osman, that we are actually on the right path by attending webinars like this. This is one right path. This is not a core specialized webinar, but there are now, thousands of webinars about informatics that actually talk directly. You see my talk. I try to run through. I've not discussed core results because I believe this is not a core technical presentation. So I now switch up, just move over. I do not discuss any result. So what I'm advising you, what you are doing, you are already on the right track by attending webinars like this. The next thing you also do is try to attend very technical webinars. You now build yourself, then you read, then you open up your scientific horizon gradually, and that is the way you move. Then very importantly, identify mentors. Try to identify mentors, people that can mentor you progressively. I think those are my advice.
Exactly, Prof. I think the most important thing is look for mentors first.
Yes.
Because
Look
mentor
for
will put you on the right direction, will point you to where you're supposed to read, will explain things to you, will tell you to apply this, to apply to that. And I think there is a WhatsApp group for Prof also who, yeah, for his mentorship and why he really posts a lot of things on WhatsApp. So you can really search out for his profile and send him an email so that you can discuss further. I think we are a little bit ahead of time, and I see a lot of good wishes from the chats. We say thank you for those that have stick with us from the beginning to the end of this webinar. And sorry if we were not able to read some of your questions, but we were able to see if they are really meaningful. We'll forward it to the speaker subsequently and will be answered on the chat. For those that send us good wishes, we say thank you very much. And as we said, this webinar is available on our social media platform. You can access it at any time and you'll be able to see the content again and again and again, and you can post question even on the go. We'll be able to look into it. For more information about BioRTC, you need to visit our website on www.biortc.com. We have a lot of interesting summer schools, engaging activities to mentor young scientists and to even support established researchers from Africa, from Nigeria. all around the world that wanted to come back home and contribute to the African science. So, on behalf of me, who moderates this webinar today, and all the BioRTC team that helped this webinar can become fruitful, and our director, Professor Mahmoud Bukamena, we extend our gratitude to our speaker, Professor Awal Ibrahim, for joining us live today, despite
Mhm.
his busy schedules. And we would like to host you again, probably at your own convenience, to discuss this paradigm shift because it's something that is really important. So, thank you very much. Wish you good evening, all, and bye.
Bye bye. Thank you.